As we all know, in order for plants to develop and grow, they need air, water, soil nutrients, and most importantly, sunlight. And just like humans, plants have different parts and functions that enable them to make their own food.
Like plastids. Plastids are double membrane-bound organelles inside plants and algae, which are primarily responsible for activities and related to making and storing food, and they are the site manufacture and storage of important chemical compounds used by the cell. When it comes to energy, generally, chlorophyll absorbs sunlight and uses its energy to synthesize carbohydrates from CO2 and water. These processes are what we call photosynthesis.
Chloroplasts are also called as the food producers of the cell. Chlorophyll is found in the thylakoid sacs of the chloroplast. Chloroplasts are a vital component of a photosynthetic cells in a plant and algae, and are unique to them. They are present in all green-colored cells of a plant, not only in leaves, but also in green stems and green parts of a fruit. A chloroplast is one of the three types of plastids, characterized by its high concentration of chlorophyll. The other two types, the leucoplast and the chromoplast, contain little chlorophyll and do not carry out photosynthesis. But what if plants can't photosynthesize due to lack of sunlight?
If a plant is kept out of light for several days, it's normal chloroplasts will actually convert into etioplasts. Etioplasts are chloroplasts that have not been exposed to light. They're usually found in flowering plants, also known angiosperms, grown in the dark. When higher plants, not all plants, are grown into darkness, they fail to form chlorophyll and develop their own plastids, a characteristic of etioplast stage. Instead of stack of thylakoids in chloroplasts, etioplasts contain paracrystalline membrane structures. It is the site of pigment protochlorophyllite, which is present in the etiolated leaf, and also of carotenoid pigments. It is in the part responsible for the arrested state of development exhibited by etioplasts. Certain thylakoid membrane proteins as well as photosynthetic pigments needed for photosynthesis are lacking.
It undergoes ethylation. It is characterized by long weak stems, smaller leaves due to longer internodes, and a pale yellow color or chlorosis. Elongation is controlled by the plant hormones called auxins, which are produced by the growing tip to maintain apical dominance. Auxins diffuses and is transported downwards from the tip with effects including suppressing growth of lateral buds. Auxins are active in light. When they are active, they stimulate proton pumps in the cell wall, which increases the acidity of the cell wall and activates expansion that weaken the cell wall and allow the cell to expand.
Due to this change, it turns into the condition of chlorosis, has little or no ability to manufacture carbohydrates through photosynthesis, and may die unless the cause of its chlorophyll insufficiency is treated. The lifespan of plants with etioplasts is based on its structural organelles and shorter than plants with normal cycle.
It's more like a downgrading system, so more disadvantages occurs.
Etiolation tends used on a small scale to treat stock plants, so the resultant cuttings are easier to root as softwood cuttings under mist propagation. This is done in early spring just as growth begins and the cuttings are handled. Researchers are still finding its usefulness and other effective purposes that will eventually give purpose leading to future discoveries.